Hello everyone. Myself Lokesh Chaudhari, preparing for NEET 2024 exam under guidance of People Fellowship. As more than half of our syllabus is completed, I'm gonna explain some of the rapid questions of NEET 23 paper of biology which you will. So we'll start with the first question. Our first question is how many ATP and NADPH2 are required for synthesis of one molecule of glucose during Kelvin cycle? We know that for one Kelvin cycle, 3 ATPs and 2 NADPH2s are used. And we know that for synthesis of one glucose molecule, 6 such cycles are performed. So we are going to multiply these both by 6. We will get 18 ATPs and 12 NADPs. Just a sec. 12 NADPH2. So second is our correct option. Next question. The phenomena of pleiotropism refers to. So we know that pleiotropy is a condition where a single gene controls two or more than two traits. So from the options, we can see that this third option matches with our definition that a single gene affecting multiple phenotypic expressions. So third is our correct option. We'll move to our next. Frequency of recombination between gene pairs on same chromosome as a measure of distance between genes to map their position on chromosome was used for the first time that. It is a straightforward question, also given in NCERT. Sir Thomas Hunt Morgan was the first person who who described who measured the distance of genes on chromosome through through gene mapping. Therefore, Thomas Hunt Morgan first option is correct. Now we got which the micronutrient is required for splitting of water molecule during photosynthesis. So splitting of water during photosynthesis has a specific word called as photolysis. Photo means light and lysis means breakdown. So here photolysis means breakdown of water in the presence of sunlight. Mm, two major elements are responsible for photolysis. They are manganese and chlorine. So here manganese is given. Therefore, first is our correct option. Next question we got. The reaction center in PS2 has an absorption maxima at. We know that there are there are two photosystems, PS1 or photosystem 1 and PS2, photosynthesis 2, photosystem 2. We know that PS1 has maximum absorption at 700 nanometer and PS2 has maximum absorption at 680 nanometer. In question, we have asked for PS second, so 680 nanometer option 1 is correct. Next question, in tissue culture experiments, leaf mesophyll cells are put in a culture medium to form callus. This phenomenon may be called as. So we will see first what callus means. So callus means it is undifferentiated unorganized proliferated mass of plant cells. Proliferated, we can also say that it is continuously growing mass of plant cells. Continuously growing means it is performing cell cycle. So the cells which leave cell cycle are differentiated cell. They got differentiated through the process of differentiation. These are continuously growing, dividing. Therefore, they have to de-differentiate themselves So that so that process is called dedifferentiation. Our second option is correct. The thickness of ozone in a column of air in the atmosphere is measured in terms of. This is a straightforward question. You have to remember that the uh, column of air in atmosphere, ozone column, is measured by the option C. Next question is an assertion reasoning type of question. Here for assertion, we got ATP is used at two steps of glycolysis. This statement is correct. Two steps means first and third step of glycolysis. And for reason, they have given first ATP is used in converting glucose into glucose 6-phosphate, and second ATP is used in conversion of fructose 6-phosphate into fructose 1,6-diphosphate. So reason is correct for the assertion. Conversion of glucose into glucose 6-phosphate is the first step of glycolysis, and conversion of fructose 6-phosphate into fructose 1,6-diphosphate is the third step of glycolysis. So assertion and reason both are correct, and reason is correct for assertion. Therefore, first option is correct. Next, in the equation GPP minus R NPP equals to NPP, uh, uh, GPP is primary productivity, NPP
Gross primary productivity.
GPP is gross primary productivity, NPP is net primary productivity. Here R is. So gross primary productivity means the total number of carbon compounds formed during the photosynthesis, and NPP is amount of carbon retained in ecosystem after the respiratory losses. So here R stands for respiratory loss. Third option is correct. Next question. An unequivocal proof that DNA is the genetic material was first proposed by. We will see unequivocal. The term unequivocal means leaving no doubts or completely explaining. So we know that Alfred Hershey and Martha Chase had conducted an experiment using bacteriophages and radioactive heavy isotopes of sulfur and phosphorus. So second option is correct. What is the role of RNA polymerase 3 in the process of transcription in eukaryotes? We know that there are three types of RNA polymerase. First is RNA polymerase 1. RNA polymerase 1 is responsible for the synthesis of rRNA or ribosomal RNA. Second is RNA polymerase 2, which is responsible for the synthesis of mRNA or messenger RNA. And third is RNA polymerase 3, which is responsible for synthesis of tRNA or small RNA such as srRNA and snRNA. So we can see that from options, second option matches, therefore it is a correct option. Next question. Among eukaryotes, replication of DNA takes place in. This is a straightforward question from the cell division chapter. DNA replication takes place in the S phase of cell division. Which of the following stages of mitosis involves division of centromere? So centromere is divided in the anaphase. As in metaphase, the chromosomes are arranged at equator. And in anaphase, they begin to move towards the poles. So during anaphase, the centromere divides. Therefore, third option is correct. In gene gun method used to introduce alien DNA in host cells, microparticles of metal are used. Microparticles of which metals are used? So we know that gene gun method is a direct method for preparation of recombinant DNA. We commonly use tungsten and gold for bombarding the microparticles in the host DNA. So here third option is correct. Next question we got, cellulose does not form blue color with iodine because the phenomenon of showing blue color is due to because some molecules like starch has helical structure which hold iodine, and after it is felt it shows blue or black color. So cellulose does not contain that helical structure, therefore it cannot hold iodine, which does not show uh, blue color. Therefore, third is our correct. Next question. The process of appearance of recombination nodules occurs at which substage of prophase 1? So recombination has another term called as crossing over. We know that in patching, crossing overs occurs between non-sister chromatids of homologous chromosome. Therefore, second patching is correct answer. Next question. Upon exposure to UV radiations, DNA stained with ethidium bromide will show. So this is a straightforward question from biotechnology. You have to remember that upon exposure to UV radiations, ethidium bromide will show bright orange color. So bright orange color is given in fourth option. Therefore, fourth is the correct answer. Next question we have got, movement and accumulation of ions across a membrane against their concentration gradient can be explained. Here, this against is the you can say it is a main term. Uh, for the accumulation of ions against their concentration gradient, ATP or energy is required. So where energy is required, the transport is called as active transport. Therefore, fourth is correct. During the purification process for recombinant DNA technology, addition of chilled ethanol precipitates out. This is also a straightforward question. We use chilled ethanol to precipitate out DNA, or we can say that after addition of ethanol, the purified DNA, we can separate purified DNA from the, we can say, extra micromolecules waste products. Therefore, chilled ethanol is used to precipitate out DNA. How many different proteins does the ribosome consist of? This too is a straightforward question. The statement is clearly mentioned in NCERT. Ribosome consists of 80 proteins. Now here we have got a match the pair questions. We have to match some steps with their characteristics or some other. So oxidative decarboxylation. Oxidative decarboxylation, it is a process which acetylation is the process of oxidative decarboxylation, where pyruvate is converted into acetyl CoA, and the enzyme conducting this reaction is pyruvate dehydrogenase. Therefore, we will match this by second. Glycolysis. Glycolysis has its other name as EMP pathway after the names of its scientists. So we have to match 3 to 4. Now oxidative phosphorylation means we reduce oxygen here and ATP is formed, which occurs during the electron transport system, as the last electron acceptor is oxygen. Tricarboxylic acid cycle, or we can say that Krebs cycle, is alternative name of Krebs cycle. Here, first step is that oxaloacetate is converted into citrate with the help of acetyl CoA, and the enzyme performing this reaction is citrate synthesis. So we'll match this. So 2, 4, 3, 1. Last is our option 4. Here too we have got the match the pairs. One side as first terms are given and we have to match definitions are given according to it. So cohesion means it is a mutual attraction among water molecules. So we will match this to this. Adhesion means the attraction towards polar surfaces or opposite charges, we can. So this goes here. Surface has more attraction in liquid phase. So we'll match these two first. And guttation is the process. Guttation is where water lost in tension liquid phase. So this was, now we got 2, 4, 1, and 3. first is the correct. Here also phases of cell division are given, and in the list two their characteristics are given. We know that in M phase, equational division occurs. M phase contains four subphases, namely prophase, metaphase, anaphase, and telophase. Here the division of cell occurs. So it is an equational division. G2 phase, this is a phase of protein synthesis which are required for cell cycle. We will match this by proteins are synthesis. Quiescent stage is a stage where cell cycle is not performed, or it is an active phase, we can say. So we'll match this by second. And G1 phase, it is an interval where interval after mitosis, means in G1 phase mitosis is completed, and after G1 phase it is S phase where DNA replication occurs. Therefore, it is interval between mitosis and initiation of DNA replication. In G1 phase, also the proteins required for DNA replication are synthesized. So, first, second, and third. These are correct.
Very good.
Next question we got. Which of the following combinations is required for chemoosmosis? We know that chemoosmosis is the process of formation of ATP through electron transport chain. So for electron transport chain, the combination required is a membrane, a proton gradient, a proton pump, and ATP synthase. ATP synthase is name of complex pi, which synthase ATP. So therefore, first is our correct option. Here we have to question is main steps in formation of recombinant DNA are given below, and we have to arrange them with correct sequence. So I will write it here. First step of formation of recombinant DNA, we have to precipitate, we have to isolate the DNA. So first step is isolation. For isolation, we use chilithanol, which purifies, means separates the purified DNA from impurities. Second stage is fragmentation, or we can say cutting of DNA with use of restriction enzymes. Commonly, restriction enzyme second is used because it cuts the DNA at the place of recognition site. Third step is amplification of DNA. Through PCR, polymerase chain reaction. And fourth, we insert the DNA into host cell. So insertion into host cell. Here isolation is labeled as C, fragmentation is labeled as B, amplification is labeled as D, and insertion of recombinant DNA in host cells is labeled as A. So we get the sequence C, P, D, and A, which is third option. Now we have to select the correct statements about Klinefelter syndrome. We'll seek statement A. This disorder was first described by Langdon Down in 1866, which is a wrong statement, as it was this disorder was described by Harry Klinefelter. Second B. Such an individual has overall masculine development. However, the feminine development is also expressed. This is a correct statement. The sex chromosomes in this such person are X, X, and Y. This is the combination of person suffering from Klinefelter syndrome. Therefore, it is masculine development because of Y chromosome, and feminine development is also expressed due to the extra X chromosome. C. The affected individual is short stature. Short stature means the person who cannot grow their height. Is there we can say genomes, not people. dwarfism. So these are short persons in simple language. It is not a symptom of Klinefelter syndrome. D statement, physical, psychomotor, and mental development is retarded. These are the symptoms of Down syndrome. So this is the incorrect statement. And E, such individuals are sterile. This is correct because of addition of an extra chromosome. Therefore, B and E statements are correct. Option third. Very good. Now, malonate inhibits the growth of pathogenic bacteria by inhibiting the activity of. This is a straightforward question. You have to remember that malonate inhibits the growth of pathogenic bacteria by inhibiting the succinic dehydrogenase. Option A. Next question we've got. Which of the following common sexually transmitted diseases is commonly curable when detected early and treated properly? We will see the options first. Genital herpes, it is a sexually transmitted disease. Gonorrhea is also a sexually transmitted disease. And HIV infection is also a sexually transmitted disease. Hepatitis B is not a sexually transmitted disease. It is a viral infection. So 3 option is not the answer. From these 3, gonorrhea is the only disease which can be cured at early stage. HIV infection is not cured, and genital herpes, we don't know the treatment at this stage. So second is correct. Next question we got. Which of the following are not considered as a part of endomembrane system? This here is the word which you have to consider. We know that endomembrane system consists of endoplasmic reticulum, Golgi apparatus, or Golgi body complex. Third is lysosome and nucleus. Lysosome. Lysosome. So we have asked that which is not considered. Mitochondria is not a part of endomembrane system. Endoplasmic reticulum is. Chloroplast is also not. Golgi apparatus is, and peroxisome is not. Therefore, A, C, and E. Second is correct option. Vital capacity of lung is. So we will see first what is vital capacity. Vital capacity is the maximum volume of air a person can inhale after an post expiration, or we can also say that the maximum amount of volume of air which a person can exhale after a post inspiration. So this formula is given in NCERT that vital capacity equals to IRV plus ERV plus TV. Here IRV refers to inspiratory residual volume, which is the total amount of total volume of air a person can inhale after a normal expiration. ERV stands for expiratory residual volume, which means that total volume of air a person can expire out or exhale out after a normal expiration. And TV means tidal volume, which is the volume as normal expiration. So fourth is correct. Next, we have got the match the pair of lack operon. On list 1, we have given the genes, and list 2, the enzymes produced by them. We know that gene A is responsible for the synthesis of transacetylase. Gene Y is responsible for the synthesis of permease. Gene I, it is an inhibitory gene which synthesis the repressor protein which can turn on or off the operon. And gene Z is responsible for the synthesis of beta-galactosidase. So second, third, fourth, and first. Second option is correct. Next question we have got. some this is CCK stands for whole cytokinin, which is secreted by pancreas. GIP refers to gastric inhibitory polypeptide, which is secreted by gastric glands. ANF stands for atrial natriuretic factor, which is from heart, and ADH stands for antidiuretic hormone, which is through kidney. So fourth, third, second, first. A correct. A is correct. Next question, we have to select the true statements among the two. For first statement, they have given low temperature preserves the enzyme in a temporary inactive state, whereas high temperature destroys the enzymatic activity because of proteins are denatured. We have to know one thing about the enzymes, that enzyme only works in optimum temperature, in specific temperature. If the temperature is lower, they can be preserved in an inactive stage, and if the temperature is increased, the proteins are denatured in them and they get destroyed. So this statement is correct. For second statement, they have given when the inhibitor closely resembles the substrate in its molecular structure and inhibits the activity of enzyme, is known as competitive inhibitor. This is a definition of competitive inhibitor. Therefore, second statement is also correct. Therefore, option A. Next question, on list 1, we have given cells, and on list 2, we have given secretions. Peptic cells, also known as chief cells, these are responsible for the secretion of proenzyme pepsinogen. Goblet cells, or we can also say that these are goblet cells, intestinal mucosal epithelial cells, which are responsible for the secretion of mucus. Oxyntic cells or parietal cells are responsible for the secretion of HCl, and intrinsic factor, which absorbs vitamin B12, and hepatic cells or liver cells, these are responsible for the secretion of bile juice. So 3, 1, 4, 2. Third is our correct. Next question we got, which one of the following techniques does not serve the purpose of early diagnosis for disease for its early treatment? There are many various techniques from which we can diagnose a disease. Urine testing, blood testing, or fecal examination, or polymerase chain reaction, ELISA, etc. are some of the diagnosis we can use. Here, serum and urine analysis, we use it for diagnosis. Polymerase chain reaction is also used, and ELISA is also used. Recombinant DNA technology is not used for diagnosis of disease. Rather, we use this recombinant DNA technology to genetically modify some plants. For example, it's a flower star tomato, which we obtain from the recombinant DNA technology method. So these are correct. Next question, which of the following functions is carried out by cytoskeleton in a cell? Major functions of cytoskeleton are: it provides motility, it helps in cell division, it provides mechanical support to cell, and we can say that specific shape of the cell is due to say cytoskeleton, so it maintains shape. So from the options we can see that motility is given here. Therefore, third option is correct. Broad palm with single palm crease is visible in person suffering from. This is this is a symptom of Down syndrome. Single palm crease means, I will show you. This is a human palm, and single palm crease start from here and end here. In normal human, the palm crease is divided into two parts. One starts from here, it cuts here, and it's continued. So the person suffering from Down syndrome, he has a single palm crease. Therefore, Down syndrome is correct. In this question also, we have to select the correct statement. Statement 1. In prokaryotes, the positively charged DNA is held with some negatively charged protein in the region called nucleoid. This statement is incorrect as the DNA is always negatively charged. They have given that DNA is positively charged, which is incorrect. So prokaryotic DNA is also negatively charged, which wrap up around positively charged proteins and form nucleus. This statement is incorrect due to positively charged. Second statement, in eukaryotes, the negatively charged DNA is wrapped around the positively charged histone enamel to form nucleus. This statement is correct. So our statement 1 is false and second is correct, which is option 4. Next question. Here in list 1, they have given the drugs, and in list 2, their effects. So heroin slows down the body function. Marijuana is responsible. Marijuana has its effect on cardiovascular system. Cocaine, it interferes with the transfer of dopamine. Transport of dopamine, so we can match it with both. And morphine is taken as a painkiller. So 2, 1, 4, 3. First option is correct. Next question. In which blood corpuscles the HIV undergoes replication and produces progeny viruses? This is also a straightforward question from Human Health and Diseases. You have to remember that HIV undergoes replication in TH cells or T helper cells. Next question. In list 1, several diseases are given, and in list 2, their positive agents are given. We know that ringworm is the causative agent of ringworm is trichophyton. Filariasis is caused by Wucheria brandipopti. Malaria's positive agent is Haemophilus influenza.
Plasmodium virus.
And sorry, is the positive agent of pneumonia. Positive agent of malaria is Plasmodium virus. The causative agent of pneumonia is Haemophilus influenza. Therefore, 2, 3, 4, and 1 A is correct. Next question, assertion reasoning question is given. For assertion, they have given nephrons are of two types, cortical and jutax medullary, based on their relative position in the cortex and medulla. So this is the structure of nephron. This is the structure. Nephron is situated in cortex. So if the Henle's loop is small, it is cortical nephron, and if it elongates till the medulla, it is a medullary nephron. So for reason they have given juxtamedullary nephron have short loops of Henle, whereas cortical nephrons have longer loop. This statement is incorrect. As shorter the loop of Henle, it is cortical nephron, and longer Henle's loop is from juxtamedullary. And assertion is correct. Therefore, assertion is true, but reason is false. Next question, we have to select the correct statement. Statement 1, a protein is imagined as a line. The left end represented by first amino acid, C terminal, and right end represented by last amino acid, N terminal. This is correct that we consider the protein as, we imagine a protein as a line. At the left end, the N group is present. It is known as N terminal. The left terminal is known as N terminal. And for the right terminal, the carboxyl group is present. So it is termed as C terminal. Here they have exchanged the positions. So statement 1 is incorrect. For statement 2, we got adult human hemoglobin consists of 4 subunits, 2 subunits of alpha type and 2 subunits of beta type. This statement is true. So statement 1 is false and statement 2 is true. This question also we have to select the correct statements. First statement, RNA mutates as faster. This statement is correct as RNA is slightly unstable. It mutates fast at faster rate. And statement second, viruses having RNA genome and shorter lifespan mutates and evolve fast. This statement is also true because the virus contains RNA, which is obviously unstable, so it gets muted faster. So both statements are correct. Option A. So here we have got a question of pedigree analysis. They have asked which one of the following symbols represent meeting between relatives in human pedigree analysis. We have to um, we have to remember some symbols here. An unshaded square stands for unaffected or it's normal or unaffected male. So normal male. Unshaded circle, it represents normal female. Shaded square represents deceased or abnormal male. And shaded circle represents the affected or deceased female. Carriers are also shown by these symbols. So first option they have they have connected the square and circle with a single lines, which represent the male and female are not closely related. So this is incorrect. Second option they have given square and circle is connected with two lines. This symbol represents the mating between relatives. So second is correct option. Next question they have given. Once the undigested and unabsorbed substances enter the cecum, their backflow is prevented by. So for prevention of backflow, there are various types of filter guarding some specific locations to prevent the backflow. Sphincter of Codi, it is present in the second section or the lower section of duodenum. Ileocecal valve, it is present at end of small intestine and start of large intestine. Gastroesophageal sphincter, it is situated at the upper part of stomach. And pyloric sphincter is present at the after the means just after end of the stomach. The last part of stomach is pyloric, so therefore this sphincter is known as pyloric sphincter. It is after the end of stomach. We have asked that a sphincter which prevents the backflow through cecum. So ileocecal valve is answered. Second one. Next question we got, which of the following is not a cloning vector? This is a simple question from biotechnology. Here, BAC stands for bacterial artificial chromosome. YAC stands for yeast artificial chromosome. PBR 322 is the first artificial cloning vector. So, fourth option is our correct. DNA probe is a sequence of DNA which is complementary to a specific genome. So, we have got the match the pair question. On list one, they are given complexes of ECG, and on list two, what they represent. ECG graph is like this. This is the P wave. This is Q, R, S, and this complex is called as QRS complex. And here is P wave. So P wave represents the depolarization of atria, which is third. Q wave represents the beginning of systole, which is first. In this QRX complex, it represents the depolarization of ventricles. So fourth, and T wave, it represents repolarization of ventricles. Second. So three or four. Option first is correct. Next question, we have to select the correct statements. Statement A, an excessive loss of body fluids from the body switches off the osmoreceptors. This is an incorrect statement because excessive loss from the body, excessive loss of body fluids from the body, it turns on the osmoreceptor. Second, ADH facilitates water reabsorption to prevent diuresis. This is a correct statement. ADH means an antidiuretic hormone secreted by kidney in the cases where blood pressure drops. So it facilitates blood reabsorption, and it tries to bring back the blood pressure to normal by increasing blood volume. This statement is correct. ANF causes vasodilation. This is also correct. ANF means atrial natriuretic factor secreted by heart in cases of high blood pressure. Vasodilation means elongation of the blood capillaries or blood vessels to easy flow of blood. So vasodilation, it increases the radius of blood vessels for decreasing the blood pressure. High blood pressure is injurious to heart and kidney. The D statement they are given, ADH causes increase in the blood pressure. This statement is also correct, as we have seen that ADH facilitates water reabsorption, which is responsible for increasing blood volume, thereby increasing the blood pressure. Statement E, ADH is responsible for decreasing GFR. This statement is incorrect. GFR stands for glomerular filtration rate. As ADH facilitates water reabsorption, the blood pressure increases, and as the blood pressure increases, the GFR also increases. They have given decreases, so it is incorrect. Here increases should be given. So B, C, D, and are correct statements. Second option. In this question too, we have to select the correct statements. For statement 1, they have given basophils are most abundant cells of total WBCs. This statement is incorrect as neutrophils are the most abundant cells of total WBC. Second statement, basophils secrete histamine, serotonin, and heparin. This statement is correct. The statement is mentioned in NCERT. Basophil is responsible for the secretion of histamine, serotonin, and heparin. C statement, basophils are involved in inflammatory responses. This statement is also correct. Basophils are neutrophils. Both are involved in inflammatory responses. Basophils have kidney-shaped nucleus. This statement is incorrect as the structure of basophil is like this. It contains a pile of nucleus. So here a kidney-shaped nucleus is incorrect. And for each statement they have given basophils are agranulocytes. This statement is also incorrect because basophil is a type of granulocytes. We can see that these are granules. So E statement is also incorrect. B and C are correct. For option third. Now we have given which one of the following is the sequence of corresponding coding strand if the sequence of mRNA is as follows. So we have given the sequence of mRNA. You have to pay attention towards this word for which strand they have asked. You have to remember that mRNA is synthesized by considering the coding strand. So here is template strand. And this is coding strand. mRNA is synthesized according to the base present on template strand. So mRNA contains base complementary to that of the template strand, and coding strand is complementary. Coding strand contains complementary bases that of template strand. So we can conclude that mRNA and coding strand have the same sequence as they both are uh, complementary to the template strand. Just the difference is that uracil is replaced by thymine in case of coding strand. The coding strand contains thymine as it is a DNA strand, and uracil, which is an RNA strand, contains sorry, mRNA is RNA which contains uracil instead of thymine. So you have to search this sequence. Just check that U is replaced with T. We can see that in option third, the sequence is correct. You have to pay attention towards the ends too, as this is 3 dash and this is 5 dash of template strand. Here mRNA is synthesized, which has 5 dash end here and 3 dash end here, which is same as coding strand. So 5 dash at the left and 3 dash at the right. Therefore, third option is correct. We have given in list 1 some cells and in list 2 their tissues. Mast cells is an example of areolar connective tissues. Inner surface of bronchioles contains the ciliated epithelium. Structure of ciliated epithelium is like this. It increases the surface area for absorption purposes. Ciliated epithelial tissues are also present in intestine, small intestine. Blood vessel is an example of specialized connective tissue, and tubular parts of nephrons, they contain the cuboidal epithelium. So 2, 1, 4, and 3. Third option is correct. In statement, we have to select the correct statements. Statement 1 we got during G0 phase of cell cycle, the cell is metabolically inactive. This statement is incorrect as they have said that G0 phase cell is inactive. In G0 phase, the cell just leaves the cell cycle. It is not metabolically inactive. It is metabolically still active. In G0 phase, it just skips the cell division.
Go for the differentiation.
Go for differentiation. Statement 2. The centrosome undergoes duplication during S phase of interphase. This statement is correct. Centrosome and DNA replication occurs in the S phase. In at very initial stage, the centrosome duplicates, and after some time in S phase, the DNA gets replicated. So second is true and first is incorrect, which is option 4. Here also we have to select the correct statements. A statement, tetrad formation is seen in leaf protein. This is incorrect. As tetrad formation is, we observe tetrad formation in zygote. During anaphase, the centromeres and chromatids split. This is true as the chromatid moves towards opposite pole. Therefore, centromere and chromatids are separate. Terminalization takes place during phageting. This is incorrect. Terminalization takes place during dikinesis, which is the last substage of prophase 1, which is incorrect. D. Nucleolus, Golgi complex, and ER are reformed during telophase. This is a true statement. All the cell organelles which disappears at the start of prophase, they reappear or reform during the telophase. Therefore, D is correct statement. And E, crossing over takes place between sister chromatids of homologous chromosomes. This statement is also incorrect, as the crossing over takes place between non-sister chromatids of homologous chromosomes. Therefore, here B and D statements are correct, which is option 2. So this was my explanation on the rapid questions of NEET 23 paper. Thank you for watching the video. Ultra.